Today we're diving into the intriguing world of neurobiology, exploring how everything from autism spectrum disorder to the types, molecular interactions paints a picture of our brain's incredible design. Let's start with autism spectrum disorder. It's a condition that affects how the brain develops and functions, often involving differences in neuronal connectivity, synaptic activity, and neuroglial support. Now these connections rely heavily on the brain support system, glial cells. Among them are astrocytes and oligodendrocytes. Astrocytes are like the brain's caretakers, regulating glutamate metabolism to keep excitatory signals in check and supporting synaptic homeostasis. Imagine them as the brain's busy traffic controllers, ensuring everything runs smoothly. Oligodendrocytes, on the other hand, are the myelin makers, creating the insulating layer around neuronal axons. Myelination is vital for efficient communication across brain regions, especially in the cortical and subcortical areas like the prefrontal cortex, amygdala, hippocampus, and striatum. What controls these glial activities? Enter adenosine, a tiny nucleoside that plays a big role. Derived from adenonucleotides, adenosine is produced and metabolized via specialized enzymes like extranucleoside triphosphate diphosphohydrolase, which breaks down ATP and other nucleotides into adenosine. This molecule can then bind to G protein-coupled adenosine receptors. Think of them as the brain's tiny switchboards, forming homoologomers or heterooligomers that influence neuronal activity. This adenosine signaling is part of the adenosinergic system, which interacts with neurotransmitters like dopamine, glutamate, serotonin, acetylcholine, and noradrenaline. These chemicals are the brain's messengers, orchestrating everything from mood and motivation to learning and memory. For instance, dopamine is like the brain's reward courier, while glutamate and GABA are the primary excitatory and inhibitory signals. Within this network, synaptic proteins and structures like dendritic spines facilitate connections between neurons. Proper synaptic pruning during development removes excess connections, sharpening neural circuits. Microglia, the brain's resident immune cells, act like gardeners, pruning unnecessary synapses and maintaining brain health. They also respond to cytokines, small signaling proteins produced during immune activation, modulating inflammation and supporting neuronal survival. Oligodendrocyte precursor cells are the progenitors that mature into oligodendrocytes, fueling myelination. On the molecular level, proteins like neuroglial support cells, cytoskeletal elements such as glycogen, synthase kinase 3 beta, and components of the beta-catenin destruction complex play a role in maintaining cellular integrity and signaling. Thanks for joining me on this world tour of the microscopic yet mighty world inside our brain.